recognized class of risk from endotoxin as an impurity, and this is immunogenicity, and it's based upon the adjuvant activity of endotoxin as an impurity. So endotoxin acts to stimulate the immune system and promotes the formation of anti-drug antibodies toward therapeutic proteins. So whereas historically we've only been concerned about fever, the rabbit paragen test originally is a fever-based test, and the bacterial endotoxin test, of course, is based on calculations relative to the occurrence of fever in rabbits. So historically we've been singularly concerned with detecting it as a paragen, but immunogenicity is a new view. It's not new to biologics manufacturers, but the relevance of endotoxins to this detection is an emerging view. We can get a better picture of this IIRMI paradigm from looking at the immunogenicity assessment for therapeutic proteins, therapeutic protein products. And in this guidance document, which was published in August of 2014, it references this IIRMI from Family and Wang in the FDA laboratories, and refers to trace levels of IIRMIs synergized direct tolerance to therapeutic proteins. So I want to just take a minute and discuss this paper. This guideline is said to be non-binding, but you can point out that the Q&A guideline is also said to be non-binding. So section 5 from page 18 describes this impurities with adjuvant activity. It says adjuvant activity can arise through multiple mechanisms, including the presence of microbial or host cell-related impurities in therapeutic protein products. There it references the paper, one of several papers more recently, and these innate immune response modulating impurities, including lipopolysaccharide, beta-glucan from yeast, flagellin, all the normal culprits of the microbial world here, including bacterial DNA, oligonucleotides. So these kind of bacterial products, it says, exert immune-enhancing activity by binding and signaling through toll-like receptors. And in the end, it says it's very important for manufacturers to minimize the types and amounts of such microbial or host cell-related impurities in therapeutic proteins. So here, in my view, for the first time officially, beginning with biologics, endotoxin is recognized not only for its pyrogenic effect, as it has been historically for decades, but also for its immune-stimulating effect. This pairing phenomenon, the clean therapeutic protein paired with the contaminant, is an effect that you only see in biologics due to the immunology of it. So if we ask, where can we more simply see where the IIRMI or adjuvant effect of endotoxin is active, we can see it in detoxified endotoxin used as an adjuvant, as an FDA-approved adjuvant, I might add, monophosphorylated. So they take salmonella endotoxin and they do a basic acid-base hydrolysis to bromophosphate, and then you have an endotoxin which is, in a sense, crippled or detoxified, as they say, but even this detoxification does not take away its ability to stimulate the immune system. So here we can see it's combined with the clean recombinant vaccine protein. Presumably the protein would be from, say, a malaria causing organism or a viral protein. So in order to stimulate the body to that, the detoxified endotoxin is added in with that vaccine. So this is a good thing in terms of stimulating immunity against vaccine proteins, but it can be seen as the very thing drug manufacturers want to avoid in their own therapeutic manufacturing, such that reduce the occurrence of immunogenicity. This is the part that, to me, provides a broader background for the LER phenomenon in terms of looking at LER as a means of detoxifying LPS. So if we have this endotoxin contaminant that's been sitting in LER-causing solution, you could certainly break that endotoxin down. Many people assume then that it's not harmful if you can't detect it in either LAL or rabbit pyrogen testing. But according to this model, of course, it can still retain its immunogenic potential, and indeed, these types of detoxification activities have occurred in the search for additional adjuvants in the vaccine world. So we can, in this way, view LER not as a singular phenomenon, but really as one of a dozen other ways of detoxifying endotoxin. Some simple ways, such as chemical inactivation, but also some common ionizing radiation. The use of surfactant SDS has been used. So there's a lot of references here, and it's interesting to me to kind of put this into context of what LER might mean. The basis for this view is the IIRMI. Of course, the body has I think it's 11 toll-like receptors that are active against all different kinds of microbial contaminants, and what these researchers showed at the FDA is when more than one contaminant is present, there's a synergistic effect, so that studies have shown that when low amount of endotoxin or low amount of bacterial DNA is present alone, they do not produce a response in mice. However, when these are combined, they become very sensitive to the presence of these things, these IIRMIs. What they demonstrated was in giving a therapeutic protein, in this case they used recombinant EPO, to the mice, so they gave recombinant EPO, and then they had another arm of the study in which they gave recombinant EPO contaminated with very low amounts of these two contaminants. And in this way, what they showed was that the promotion of immunogenicity from the contaminants was able to contribute to what they call a clinically relevant break in tolerance to self. So, in simpler terms, the recombinant protein only produced a transient change in the control study mice, but in the mice with the contaminant, it produced a long-lasting anemia, which told the researchers that this therapeutic protein with contaminant not only produced antibodies against the recombinant EPO, but it also began attacking the rodent's naturally occurring EPO. So I would encourage you to look at this study if you have an interest in. Also, some of the further references listed in the immunogenicity assessment guideline. So detoxification here is a structural change to what you see on the right-hand side. There's the normally occurring lipid A as seen in E. coli with several different sites that would be detoxified or that would be changed in order to change the ability of the endotoxin to produce a pyrogenic response. So in the case of NPLA, the acid hydrolysis removes the phosphate and makes it 100 to 2,000 times less pyrogenic, depending on the method of detoxification. And this kind of shows us the dual nature of endotoxin as pyrogenic and also as immunogenic, even when it is not pyrogenic. It's also interesting to me to ask, what does the body evolve to do with various endotoxin? Of course, the human body and other mammalian bodies are complex systems. They're not simplistic like the horseshoe crab. It basically has one reservoir of blood that consists of its whole body. The human system is partitioned and contains a surprising number of bacteria in the gut that have to be kept out of the tissues and the sterile blood. So we can see, in effect, what the body's trying to do is to keep out organisms from the environment, but also, and seemingly more importantly, it wants to keep in those most potent types such as E. coli that inhabit the gut and mucosal linings. So those specific endotoxins have these secondary acyl chains, and as we see in the E. coli standard, these produce the most potent endotoxin. So the more that science seems to learn about endotoxin, it sees that the body is not ignoring the non-extensal or the non-prototypical E. coli endotoxin. It's just not in the